Hello, welcome to the Aspirational Neuroscience Journal Club. This time we are covering this 2019 paper from Park et al. Entitled Contribution of Apical and Basal Dendrites to Orientation Encoding in Mouse V1 Layer 2-3 Pyramidal Neurons. This is a really fascinating paper that uses a brute force technique that literally chops off parts of a living neuron to see how its tuning is affected. By using laser microdissection to chop off individual dendritic branches, the authors go beyond existing literature that mainly records what information is available to, for example, the apical and basal dendrites. Instead, they are able to ask whether the neuron is actually using the information in a given experimental context. The authors are studying pyramidal neurons in layer 2-3 of the primary visual cortex of the mouse. Let's quickly review, in an overly simplified model, the receptive field tuning properties of these neurons, and review the receptive field properties of the cells that send them synaptic inputs. This is a figure from the textbook Principles of Neuroscience, showing the layered structure of the primary visual cortex in mammals. Here is a layer 2-3 pyramidal cell body, its basal dendrites in orange, and its apical dendrite in green. Axons from the lateral geniculate nucleus typically innervate layer 4, which is too low to directly synapse onto our layer 2-3 pyramidal cell. Instead, they typically synapse onto layer 4 interneurons, whose axons then project to the basal dendrites of our layer 2-3 cell. The lateral geniculate cells have receptive fields that are not tuned to edges. Instead, they are tuned to center-surround contrast. The layer 4 interneurons sum inputs from many of these LGN cells, whose receptive field locations are strung along a line. This makes these layer 4, quote, simple cells responsive to edges at a particular location and orientation. Now, our layer 2-3 cell then collects inputs from many of these layer 4 simple cells, whose receptive fields are offsets from each other spatially. This makes these layer 2-3 cells also tuned to oriented edges, but in a more translation-invariant manner. Because of this, they have been termed complex cells. This picture suggests that the main synaptic input to our layer 2-3 neuron will be from layer 4 cells, which are tuned to edges at particular orientations, and that these will synapse mainly onto basal dendrites of our layer 2-3 cell. Input onto the apical dendrites has been suggested to come mainly from higher order cells carrying feedback information. But there are many additional projections that are known to convey orientation selective information to the apical dendrites. So prior to the current paper, it was unclear how these apical and basal dendritic inputs were integrated together to give the neuron's overall direction selective tuning response. The author's brilliant solution to clear up these questions was to simply chop off dendritic branches and see what happens. Before we jump into the results, I want to make sure everyone understands just how many synapses are being chopped off in these experiments. Here is a visual cortex pyramidal neuron reconstructed in all of its glory from Ellen Institute's electron microscopy project. We can see that each of its dendritic branches has thousands of synapses. This paper literally chops off whole dendritic branches of living neurons, meaning thousands of synapses eliminated. Amazingly, the cell often survives this, and its tuning is only slightly affected. Now let's talk about the methods. The authors put the mouse under anesthesia and drill a 3 mm diameter hole in the skull over the visual cortex. The authors inject tiny volumes of 2 viral constructs, CAMP-kinase 2 Cre and a Cre-driven GCAMP6 AAV. GCAMP6 is a calcium indicator that shows an increase in brightness when the neuron has a high firing rate. The authors used this GCAMP6 fluorescence signal to measure the response of the target cell in all experiments. During the actual experiments, the mouse was sedated. This is important because it means that behavior-based feedback onto the neuron was not present during these experiments. Full field moving gratings at a range of angles were shown to the mouse's right eye, while the neuron's GCAMP6 intensity at the cell body was measured via a fluorescence microscope. It's important to understand that the 2-photon imaging that the authors performed allowed them to visualize the full 3D structure of each recorded neuron. Here is a video from the paper that shows a stack of fluorescence images. And this is the 3D reconstruction of that same neuron made from tracing this stack. This is the soma of the neuron, where the main GCAMP6 signal is recorded to measure the neuron's overall tuning. And these are the basal dendrites. The authors state that each neuron has on average 6 of these basal dendrites. In total, they receive approximately 60% of the synaptic input onto the neuron. And this is the apical dendrite that receives the rest of the synaptic input. This plot here shows one experiment, one neuron in one mouse, where the change in GCAMP6 fluorescence was measured. This plot shows the response of this one neuron to moving gratings at 12 different angles. If we zoom in on one of the subplots showing the response to vertical gratings moving to the right, we see that it is composed of 20 individual trials, each 2 seconds long, which show how the firing rate of the neuron increased to the visual stimulus. The black curve here shows how this neuron's firing rate typically increases during each showing of the vertical grating. In contrast, the neuron showed almost no response to a horizontal grating moving upward. The overall orientation tuning of this cell is plotted here. It shows a peak firing rate around 90 degrees. Now the cool part is that the authors could focus the laser scanning 2-photon microscope on a particular part of the neuron to cut off a branch. Here I'm depicting the laser cutting off the entire apical dendrite. The cut membrane quickly reseals, but the apical dendrite is no longer attached to the cell body, and as a result, it blebs and disintegrates over the next few hours. The authors can see this in the microscope, so they are 100% sure that the synaptic inputs from that particular branch have been completely removed. Here is an actual image before and after cutting off the apical dendrite of the neuron. Not all neurons that had their branches cut off survived the process, but around half did and recovered to show normal electrophysiological responses by the next day. This brings us to the first key result of the paper. The visual tuning of the cell is essentially unchanged after the entire apical dendrite is removed. Here is the response of the neuron to visual gratings at different angles on day 0, that is prior to cutting off the apical dendrite. And here is the response of the same neuron on day 1, 3, and 5 after cutting off the apical dendrite. Amazingly, the response is statistically unchanged, despite the fact that the cutting removed 40% of the neuron's synaptic inputs. The authors performed this apical dendrite ablation on a total of 18 neurons. Each of these experiments is shown as a blue circle on this plot, where the x-axis is the before removal preferred orientation, and the y-axis is the preferred orientation 5 days after the apical dendrite was removed. You can see that they fall along the x equals y line, indicating almost no change. The authors also imaged 17 control neurons 5 days apart, but with no laser ablation. These are the black squares. They show the expected variability of tuning of a normal neuron when measured after 5 days. Statistically, there is no difference between these two groups. The authors also compared the width of the tuning curves before and after ablation, and measured something called the orientation selectivity index, which is a measure of how strong the preferred response is. And they found that ablation of the apical dendrite did not change these other tuning properties either. This is perhaps the key result of the paper, so we should take a moment to unpack it. Previous experiments had led to theories that the apical dendrites of layer 2-3 neurons are mainly receiving feedback information from later stages in the visual stream, and that the basic feed-forward response of these neurons should instead come from input to the basal dendrites. The current experiment used a sedated mouse, so it is likely that feedback from later areas was minimal. Further, the visual input was a bright, high-contrast, full-field grating stimulus, a very strong feed-forward drive. So under the theory that apical dendrites are mostly for feedback, we should expect removal of these feedback inputs should produce little effect, which is exactly what the authors found. This supports the theory, but certainly does not imply that the apical dendrite is unimportant to normal processing. If the current experiments had been done in an awake behaving animal engaged in, for example, a 2 alternative force choice task with low contrast stimuli, then top-down expectations provided to the apical dendrites might have been crucial to each neuron's tuning response. Let's move on to the next key result. The authors then conducted a set of experiments where they cut off either one or two basal dendrites. Recall that each neuron only has an average of 6 basal dendrites, so each one that is cut off removes approximately 10% of the total synapses of the neuron and 1/6 of the basal synaptic inputs. Here is an example experiment where one basal dendrite was removed at the arrowhead, and these are orientation tuning plots of 3 separate neurons showing tuning before removal in black and 5 days after removal in dark red. As you can see, removing a single basal dendrite had essentially no effect on the overall tuning of the cells. And here's another experiment where 2 basal dendrites were removed at the arrowheads. And these are orientation tuning plots of 3 separate neurons, with each having 2 of their basal dendrites removed, showing tuning before removal in black and 5 days after removal in pink. Now we can see that there is a shift in tuning, but it is only on the order of 10 degrees of shift in the neuron's preferred grading orientation. The authors performed single basal dendrite ablation on a total of 13 neurons and 2 basal dendrite ablation on 17 neurons. They also analyzed data from 35 control neurons that were recorded in separate sessions 5 days apart, again to quantify the repeatability of the tuning across that interval of time. These plots summarize these experiments. The average change in orientation tuning in the control neurons between sessions was less than 5 degrees, and the change seen in the single basal dendrite removal experiments was not significantly different from that. There was a 10-degree on average shift in orientation tuning when 2 basal dendrites were removed, which was significantly different. In case you're wondering, they tried removing 3 basal dendrites, but the cells almost always died when that was tried. The width of the orientation tuning curves and the orientation selectivity index was not significantly changed relative to controls for either condition. The authors used a biophysically realistic compartmental model neuron to help make sense of these results. The model was implemented in the neuron simulation language and included realistic active dendritic conductances, so that the model would at least have the potential of amplifying clustered inputs that converged onto the same dendritic branch. Their basic assumption was that all incoming synapses were themselves tuned for orientation, but their preferred orientation was randomly chosen from a normal distribution with a particular mean and standard deviation. This assumption was chosen to agree with existing literature on the synaptic inputs to layer 2-3 neurons. For example, this drawing shows a model where the apical dendrite had, say, 4,000 synapses, each of which had its orientation preference drawn from a normal distribution with mean of 0 degrees and a standard deviation of 30 degrees. And this particular basal dendrite would have had on the order of 1,200 synapses, each of which had its orientation preference drawn from a normal distribution with a mean of 15 degrees and a standard deviation of 30 degrees. The authors found that their experimental results were best fit if each of the basal dendrites had different mean orientation preferences. For example, minus 30 degrees, minus 15, 0, plus 15, and plus 30 degrees, where the mean of all the basal dendrite preferences was the same as the apical dendrite's preference. This was needed to explain why removing the apical dendrite does not change the cell's overall orientation preference, while also explaining why removing 2 of the basal dendrites would result in a slight offset of the cell's overall orientation preference. This is the central modeling assertion of the paper. The basal dendrites must each have been independently tuned for oriented edges, resulting in a range of different orientation preferences. Reading from their summary, these simulations suggest that to explain our experimental data, there needs to be a degree of heterogeneity of tuning across basal dendrites. To summarize, here are what I see as the key takeaways. There is tremendous redundancy and robustness in even single neuron computations. Quoting from the paper, the remarkable robustness of orientation preference under dendritic microdissection hints at the extraordinary ability of sensory cortical neurons to maintain functional selectivity following input loss. And this paper shows yet more evidence for active dendrites acting as computational subunits of neurons. Quoting from the paper, to explain the small but significant post-ablation change in orientation preference following two basal dendrite ablation, simulations suggest that orientation tuning is somewhat heterogeneous across basal dendrites. Such heterogeneity may arise from dendrite-specific forms of plasticity, potentially mediated by spatially restricted biochemical signaling and/or dendritic spikes. And I want to end by reviewing what I see as the biggest limitation of the paper. The paper only measured orientation preference, tuning width, and orientation selectivity index. From the literature, we know that these layer 2-3 complex cells have spatially larger receptive fields than the layer 4 simple cells that they receive input from, and they are more translation invariant than the layer 4 simple cells. Because the authors utilized full field grading stimuli, they could not tell whether removal of dendritic branches affected these important computational properties. In my opinion, it's a good bet that chopping off basal dendrites would indeed have resulted in shrunken receptive field size and partial loss of translation invariance. I hope the authors follow up this current study with one that explores these issues. Nonetheless, I think that this paper highlights a fantastic new technique that can address what role individual dendrites play in neuronal computation. Thank you for listening.